Hi everyone, this is part 2 of the Chapter 5 lecture notes, and we had finished this slide, but I think my last few words got cut off. So just making sure you understand the role of cholesterol in the fluidity of the plasma membrane. Cholesterol, it depends really on the temperature. So if the temperature is cold, cholesterol, which is a lipid, helps to keep the membranes fluid. But if the temperature is hot, if it's a climate where the temperature is hot, the plasma membranes of organisms use cholesterol to keep their plasma membranes a little bit less fluid. It keeps them from getting too fluid when it's hot. So it just depends on the temperature as to the role of cholesterol. And this is a video I would highly encourage you to watch. Let me see what slide it is. It is slide number 12, and it is a video on membrane fluidity. And sometimes I can try to explain things with analogies about marbles and that kind of thing, but this is a really good video to watch, so I encourage you to watch this. Slide number 12. And now we're going to go on to the rest of our lecture. Plasma membranes are asymmetric. Oh, and there was another, I'm sorry, there was another term. You know what? Let me go back. Amphiphilic just made me remember this. Amphiphilic. Here we go. In okay, phospholipids are amphiphilic because they contain a hydrophilic head and a hydrophobic tail. So that's what amphiphilic is. It means it's got a hydrophilic end and a hydrophobic end. Now let's go back to, this is another, of course, a repeat of the fluid mosaic model. Plasma membranes are asymmetric. The inner surface is different from the outer surface. For example, interior proteins anchor fibers of the cytoskeleton to the membrane. So you can see here, these are cytoskeletal fibers. And let me find one that's attached to a protein. Here's one. So these fibers of the cytoskeleton are anchored by these membrane proteins. So basically that anchors the phospholipid to the interior of the cell. Exterior proteins bind to the extracellular matrix. In other words, whatever is on the outside surface of the cell, the exterior proteins will bind to that material, whether it is another plasma membrane or whether it is some chemical complex in the tissue fluid or in the tissue itself. Most cells are connected to other cells, at least on one surface or more. So proteins on the outside bind these cells to other cells or to the extracellular matrix is just, it's just like in connective tissue such as blood, the matrix is the blood plasma, the fluid part of the blood. It's just whatever chemical or material surrounds the cells in that tissue. And then glycoproteins, not glucoproteins, this should be glyco. Glycoproteins bind substances to substances the cell needs to import. The plasma membrane is selectively permeable, or you may have heard it referred to as semi-permeable. So that means it allows some molecules to pass across it, either into or out of the cell, but other molecules it doesn't allow to pass. So this feature allows the cytosol to differ from the solution outside the cell, the extracellular fluids. For example, all cells maintain, and this is every cell, maintains an imbalance of sodium and potassium. So the amount of sodium outside of a cell is high, and the amount of potassium inside the cell is high. So we have high sodium outside, and we have high potassium inside, implying that the sodium inside the cell is low and the potassium outside the cell is low. So it's high, the sodium is higher outside and the potassium is higher inside the cell. Transport across the plasma membrane can be passive. That means it does not require energy. Diffusion, for example, is a passive process. Or it can be active. That means it does require energy. Passive transport is the simplest type, the simplest type of passive transport is diffusion. There are other examples, but the simplest type is diffusion. Diffusion occurs even in other instances besides when we're talking about cells. If I spray perfume in one area of the classroom, for example, the perfume is going to diffuse all the way to all areas of the classroom. So the students on the front row will smell it first, but then the students on the back row may start to smell it after a certain amount of time, because the molecules are going to diffuse until they're equally dispersed throughout the classroom. So the definition of diffusion is when substances move from an area of high concentration to low concentration down its concentration gradient. Now, a concentration gradient is just a concentration difference. So if you have an area where the concentration of a substance is high and an area where it's low, its concentration gradient is just the concentration difference. So a substance is going to move from high concentration to low concentration. In cells, diffusion occurs directly through the phospholipid bilayer. Some molecules that can do this, any molecules that are able to diffuse through the phospholipid bilayer are going to have to be nonpolar, and they're going to have to be small. So oxygen, carbon dioxide, these are really written with the 2 as a subscript. Oxygen, carbon dioxide, and small lipid hormones can diffuse directly through the phospholipid bilayer. And you can see an example where the concentration of this substance here, it doesn't say what the substance is, but we can assume that it's small and it's nonpolar. So it's insoluble in water, but it's soluble in lipids. This substance can diffuse directly through the phospholipid bilayer from where its concentration is high to where its concentration is low. That is what we mean when we say down. That doesn't look like a W. Okay, when we say down its concentration gradient. Concentration gradient is concentration difference. And then you're going to have diffusion until equilibrium is reached. So the concentration is the same on either side of the membrane. Factors that affect diffusion rates. All right, the higher the concentration gradient, the faster the diffusion rate. So the greater the difference in concentration, the faster diffusion will be. The mass of the molecules. Smaller molecules are faster. Temperature. The higher the temperature, the faster the diffusion rate. The density of the solvent. So the solvent is what the solutes are dissolved in. Water is often the solvent, and the density of the density of the solvent, if it's less concentrated, more watery, then it's going to be less. It'll have less solvent density. So dehydration increases the density of the cytoplasm, and the more dense that the solvent is, the slower the diffusion rate. So increased density of the solvent means decreased diffusion rate. Basically, the less concentrated that your solvent is, the more watery that it is, the faster the diffusion rate is going to be. Think about a molecule trying to diffuse through syrup versus trying to diffuse through water. It's going to diffuse much slower through syrup. Solubility, the more nonpolar that materials are, nonpolar materials tend to diffuse faster because they can diffuse directly through the phospholipid bilayer. The surface area. So one reason that we chew our food up is because that increases the surface area. The smaller the food particles are, the more surface area that there is for enzymes to react and break those food particles down into smaller molecules. So increased surface area speeds up diffusion rates. That means breaking molecules down into smaller molecules, physically breaking them down, is going to speed up diffusion rates. The distance traveled, the greater the distance, the slower the diffusion. So that is one important reason why cells are so small, so that diffusion can occur faster in smaller cells. And then pressure. Kidneys have a pressure called blood pressure actually, that affects their filtration rate, how fast they can filter the blood. So the higher the pressure, the faster the diffusion rate. The higher the blood pressure, the faster the diffusion rate. Now, molecules that are nonpolar and lipid soluble can diffuse directly across the phospholipid bilayer. But other molecules which are polar or charged have to be transported through one of the membrane proteins. So facilitated transport or facilitated diffusion, which mean exactly the same thing. To facilitate means to aid or help. That means a carrier protein, one of these blue proteins here, either a protein channel or one that's what we call a carrier protein that actually carries or transports a substance across the membrane. Some type of integral membrane protein is going to assist the substance in crossing the membrane. So if the substance is charged, if it's an ion, that means it has a charge, positive or negative. And if it's small and polar, because water is polar, so any polar molecules are going to use facilitated transport for diffusion. They're going to use either a channel protein or a carrier protein. Channel proteins have a pore so that the substance can simply diffuse through that pore or that channel. Some channel proteins are open all the time, others are gated. They have a gate that requires some kind of signal to open that gate. Aquaporins are specifically channel proteins for water. And muscle cells have gated ion channels that when they're opened, they cause ions to flow through, and those ions flowing through will cause the muscle to contract. So channel proteins allow ions or polar molecules to move across the membrane by diffusion from high to low concentration.